All right, welcome everybody. Thank you so much for being here. As you know, if you've been in this class, and several of you have been in this class for a couple semesters, this is a date that I've had circled on my calendar for a long time. And as I read this introduction and as you get to know our speaker today, Dr. Gregory Prince, you'll see why. This is a remarkable individual, and we are very fortunate to have him here. I want to acknowledge first the Tanner Center for Human Values. We have Dr. Danielle Dubrasky and Michelle Lambert. They are co-sponsors of this event. And we do have a couple spots left for lunch if you're interested.
Yeah, myself and Michelle will be back here, and we can let you know if you give us your name, first come, first serve, so we could probably have a few people. Yeah.
Okay, thank you. Okay, so our speaker today is Dr. Gregory Prince. He graduated from Dixie. He then went to UCLA where he got a DDS, that's a doctorate of dental surgery, and shortly thereafter he got a PhD in pathology. He then did a postdoc with the National Institutes of Health, and then he spent 10 years at Johns Hopkins University. He then founded Biron Systems, which is a biotech company that is focused on the prevention and treatment of pediatric infectious illnesses, and in particular, RSV. Biron System produced a drug that is given to roughly 250,000 children around the world every year to prevent their death from pediatric pneumonia. He's currently CEO of SoftCell, which is a relatively new biotech company. His other accomplishments include establishing the Madison House Autism Foundation, which helps adults and families with autism deal with autism. He has written over 150 scientific papers, and he has written 5 books. Please welcome Dr. Gregory Prince.
And none of it was intentional. I thought when I enrolled at Dixie I would be practicing dentistry in a private setting the rest of my life, which is what my father and about a dozen other extended family members did. So keep your options open. That's going to be one of the messages today. This is a story of a virus that's the primary cause of infant pneumonia throughout the world. A rat you've never heard of. You probably heard of the Birds of North America that John Audubon painted. After he finished that, he did a second book with the very forgettable title of Viviparous Quadrupeds of North America. One of those paintings was the cotton rat, and the cotton rat turns out to be the central character in this drama. Another figure in it, indirectly, Dr. Jonas Salk. The Salk polio vaccine was approved by the FDA in 1955. I can remember in 1956 visiting my grandparents in St. George, going down to the city park, and right there in the park getting the Salk polio vaccine. Bad science along the way. I'll talk about that some more. The Russian connection, a message that they inadvertently sent that everybody else ignored, but we didn't. And a willingness to swim against the current. I think I've maintained that willingness for the rest of my life. 1939, a paper was published by Charles Armstrong, who was a scientist at what was then the National Institute of Health. There was only one at that time. Polio was not known in the New World until the last decade of the 19th century. And then throughout the 20th century, there were periodic waves of epidemics that were terrifying. One in 1937 was particularly bad. A young man who died of polio in Michigan became the donor of spinal cord tissue, which was sent to Dr. Armstrong. He sent his assistants throughout the southeast of the United States, basically saying, bring back any live animals that you can trap and let's see if any of them are susceptible to polio. Of this menagerie of wild animals that he brought back, there was one animal that was susceptible, and it was the cotton rat. 1948, because of the cotton rat, the very first polio vaccine was developed, but it's one you've never heard of. Hilary Koprowski was the developer of it. He was a brilliant Polish physician and musician, graduated in the same year from medical school in Rome and the Warsaw Conservatory. He was a concert pianist of Chopin. I'm going to read you from his biography. It is toward the end of a cold January day in 1948 at the huge modern complex that is Letterly Laboratories in Pearl River, New York. Winter darkness has fallen beyond the windows of the lab where research scientist Dr. Hilary Koprowski and his assistant Thomas Norton are at work. It is a simple wearing blender over which Koprowski and Norton are bending, measuring out the ingredients of a very special cocktail. And a gruesome cocktail it promises to be. The two men spoon bits of grisly cotton rat spinal cord and brain tissue into the blender. Cold, greasy stuff that has been infected with a particular strain of poliomyelitis virus. Thomas Norton, a tall, stooped man, flips on the blender. He and Koprowski frown at the machine as it whirs with a shrill scream, chunking now and then as the blades reduce chunks of solid matter to puree. Norton reaches out a hand to steady the blender so it won't vibrate its way off the stainless steel table. After it has run smoothly for a moment, he switches it off. Koprowski carefully pours 1 cubic centimeter of the cold, grayish suspension into each of 2 small graduated glass beakers. It is an oily glop. Brains are full of fat. Norton and Koprowski regard each other over the rims of the beakers. They drink, tipping their heads back for a long moment until the thick stuff flows sluggishly across their tongues. They make bad faces as they suck the greasy soup out of the corners of their mouths and swallow repeatedly. Koprowski notes that the raw vaccine tastes like cod liver oil. When he can speak, Norton asks, "Have another?" "Better not," Koprowski says. "I'm driving." But that was the first effective polio vaccine. It was never utilized in this country, but over 1 million doses were given in Koprowski's native Poland. Now we go up 7 years to when the first vaccine is approved, 1955. The way that Salk prepared the vaccine was to grow poliovirus in human cell culture, do a crude purification of the virus, add a small amount of formaldehyde, which killed the virus but didn't change the conformation sufficiently that the immune system could not recognize it, and that became the vaccine. That is a technique that has been used repeatedly. The following year, RSV was discovered at Johns Hopkins Hospital in Baltimore, discovered by the man who I later worked for, Robert Chanik. Thinking that Salk had the universal vaccine recipe, what Bob Chanik did then was to do the same thing that Jonas Salk had done, which was to raise virus in human cell culture, to add a small amount of formaldehyde to it to kill the virus, and that became the vaccine. In 1962, that experimental vaccine went into clinical trials in a very small trial that didn't have a control group, something that would be unthinkable by today's standards, as would taking a wearing blender and having an experimental vaccine that the scientist used himself as a guinea pig for. There were danger signs after this trial. I talked to one of the physicians who was involved in it. She said, "I went to Dr. Chanik and said something is wrong with that vaccine, but he wouldn't listen." And because they didn't have a control group, they didn't have any means of comparing what was happening to that group to anything else. So 3 years later, a larger clinical trial using the same recipe. The lack of a small animal model, which is the key to this whole story, precluded having any safety testing before they took this into clinical trials. 1967, January 1st, is a day that some of those researchers will never forget, because that was the day when one of 2 children who were involved in this study died. The second one died on January 2nd, 1967. The hospitalization rate among those infants that had received the vaccine and were naturally infected with RSV was 16 times higher than in the control group. If you talk to any pediatrician today, that person will still say, yeah, I know about that vaccine trial. It put a cloud over vaccinology for decades, and now here we are 60 years later, we still do not have a vaccine against RSV. The drug that we developed is replacing the immune system, it's not stimulating it. 1969, 4 papers were published simultaneously in the American Journal of Epidemiology, and they showed that the infants who had received the vaccine developed antibody to RSV. Duh, isn't that what a vaccine's supposed to do? Therefore, they said, in our view, serum antibody probably is an active participant in the process of tissue damage. It was an ill-constructed clinical trial followed by a very bad interpretation of the results, and it set the field in the wrong direction for several years. That became medical dogma, that for RSV, antibody is harmful. 1971, I began a 1-year pathology residency at UCLA. My mentor, who wrote this book later, based in large part on his experiences with virion systems, I did several autopsies under Dr. Porter during that year. Literally over a dead body, he said, "You look like you have an aptitude for research. If you're interested, after you get your dental degree, come back." I went back, finished dental school, then knocked on his door, and he said, "I've been expecting you." His mantra, and I want you to remember this if you remember nothing else that I say in this lecture, is work on something important. It doesn't matter whether it's science or any other avenue of your life. I know a lot of people who have the capability not only to work on something important, but probably to make a difference that would have a ripple effect locally or nationally or internationally, and they choose instead to spend their entire career in some backwater area and just guard their turf for the rest of their careers. In his case, he said, I was put on to elution disease of mink. It's a persistent virus infection. He said it was interesting, but it wasn't particularly important. So he said, you're going to work on RSV because it's the most important virus affecting human infants. And if you ever accomplish anything in it, there's the possibility that that will make a difference. 1973 is when I began my doctorate work under Dr. Porter's direction. There was no small animal model then for RSV, and he knew that in order to try to decipher how did this vaccine backfire, we had to have a small animal model in which we could reproduce the primary disease and then hope that we could reproduce the enhanced disease that the vaccine had caused. Turns out that we got the animal model. We never quite figured out how the enhanced vaccine worked, but we were able to go down an unintended pathway that turned out to be pretty productive. 1973 to 1975 was when I did my doctorate studies at UCLA. This was at a time before there were computer indices, databases where you could search the medical literature. There used to be a publication called Index Medicus, and every year the print version of it would get longer and longer and longer. I can remember at one point 20 linear feet of shelf space for that year's edition of Index Medicus. RSV was not even a topic yet in Index Medicus, so I had to look through the larger heading of respiratory viruses and then go through manually, take my index cards, copy down what seemed to be appropriate, and I did a deep, deep dig into the medical literature for most of those 2 years. This was the key paper that I found. It was published in Russian in a Soviet journal. The title was translated into English, and a brief abstract was translated into English. The rest of the paper was in Russian. But you can see by that title, Experimental RS Virus Infection of Cotton Rats, a Viral and Immunofluorescent Study, caught my attention. Crucial question was, could we trust the Russians? Now, there are rankings of medical journals. One of the highest ranking is the New England Journal of Medicine. You're all familiar with that title. The impact score, which is how much of an influence does what's published in this journal have on the entire field of medicine and biomedicine, is very high, 91.2. It ranks number 3 in all of the published journals. Problems in virology, the Soviet journal, look at the impact score. It barely gets off the floor, and it ranks 12,000th in all of these. And yet, we paid attention. We did have a secret weapon. It turns out that Dr. Porter's father was Nicholas Bobrovnikov. Porter said, I was born in Ohio in the 1930s. Having a Russian surname was not advantageous in those years. So I took my mother's surname. Dr. Bobrovnikov made a handwritten translation of this for us at a time when the UCLA library didn't have the capability of doing such a translation. And that turned out to be the key. So we said, we believe what these guys are doing over there. It was the only paper they ever published on it. Our next step was to try to find cotton rats. They are native to the New World, but to try to find a colony eluded us. We had some live trapped in North Carolina, sent to UCLA, couldn't get them to breed. When I went to NIH, I found out that they had a colony on campus that they had established for a parasitologist, but it had taken so long to establish it that he'd moved on and lost interest, and they thought, we'll keep it in case somebody else comes along who is interested. We were interested. This is the geographic distribution. For most of that region, this is an agricultural pest. For us, it was the key to unraveling a very serious disease. 1978, I published the model paper of this, the American Journal of Pathology. And then I started to go the next step. The first step in doing that paper was, can we grow the virus? What are the kinetics? What do the lungs and the nose look like under the microscope over the course of infection? Very cut and dried work. I'd had some exposure prior to this to the topic of liver regeneration. And during that exposure had learned about a technique called parabiosis that physiologists had used for almost a century. What they do is to do a surgical linkage of two small animals in such a way that when the surgical wound heals, the blood circulates between one animal and another. The advantage of this is that you can then do a manipulation on one animal, and if you can see an effect on the other animal, you know that that is going through the whole blood, and it gives you a leg up on trying to isolate what that active factor is. Nobody had ever used this to study immunity, but since I knew about the technique, I thought, let's give it a try. Now let me walk you through this graph. What I did was to make three kinds of pairings of these animals. In the first instance on the left, surgically linking an animal that had been infected and had recovered to another animal that had been infected and had recovered. The second was two animals that had never seen the virus that have been now connected surgically. The third and the important group was an animal that had recovered from the virus connected to an animal that had never seen it. Do the surgery, let the wounds heal, allow the blood to cross-circulate for a couple of weeks, and then infect both animals in each pairing and see what happened. The red represents the amount of virus in the lungs, which is the really important organ for RSV. You can deal with a runny nose. Dealing with pneumonia is a lot more difficult. So the immune animals remained immune in both tissues. No surprise there. The control animals were susceptible in both tissues. No surprise there. What surprised us was the third group, that we saw complete protection from pulmonary infection in the control animals and no protection from nasal infection. This went totally against the dogma that we'd been hearing about and that we had assumed was correct. We weren't doing this to try to challenge the dogma. We were just setting up experiments to say what's going on in immunity and what can we learn from it. So the next thing we did was we took whole blood, fractionated it, and found out the activity was in the fluid phase. Turns out to be antibody, which is the major protein compound of the fluid phase. And then we did what's called a scattergram. Each one of those dots represents an individual animal. We looked at the effect of increasing doses of antibody upon the amount of virus that would grow either in the lungs, which are the red dots, or the noses, which are the yellow dots. And what we saw, or excuse me, nose on the right side, the yellow dots are the control animals. What we saw was that it's dose dependent. And we could draw a line and say, if we give this much antibody, we can protect the lungs. This was the key paper that we published in 1985. But then the question became, do we follow the conventional wisdom that antibody to RSV is harmful, or do we follow the data? And my mantra then and ever since, and it's not just for science, is follow the data. It's never gotten me wrong. 1987, some of the program officers at NIH had paid attention to what we were doing. I was working still in a lab at NIH, and said, we are going to put together an RFP, a request for proposal. This is to solicit responses to a contract to test this hypothesis in a clinical trial. We put together a consortium of 3 medical centers, bid on the contract, won it, and started a clinical trial of purified human immunoglobulin to see if it would work in humans the way it had in the animals. 2 years later, after our phase 1 clinical trial, we informed a strategic alliance with a biotech startup company called MedImmune that later became a really big biotech company and eventually was purchased by AstraZeneca. 1996, all of this is a lot of years, and yet by industry standards, it was pretty quick. We had our first generation product. It was called Respigam. It was derived from human plasma, and it worked. Problems with Respigam are that each lot of the drug required 10,000 units of plasma, 1 unit from 1 patient donation, that only 5% of the potential donors had high enough levels to qualify to be donors. So you do all of that calculation and say, wow, it's 200,000 potential patients to get 1 lot of plasma. And then the volume of the drug necessary, because we were just taking all of the antibody, purifying it, and giving it to the infants. It was whatever antibody they had, and most of it was not RSV. You had to immobilize the patient for up to 4 hours at a time once a month. And with these preemies, trying to get intravenous access is a real problem. But it worked, and it got us into the game. The solution long-term was, let's make a monoclonal antibody. Now, if you pay attention on many of the TV stations, much of the advertising that you see is pharmaceutical companies advertising drugs for chronic diseases, and if any of those drugs, the generic name you see MAB on the end of it, that is a monoclonal antibody. These are miracle drugs that are being used for cancer, for chronic diseases, but up until now, there's only 1 monoclonal antibody that's ever been given full licensure by the FDA for any infectious disease, and that's the one that we developed. So what is a monoclonal antibody? You have two main types of immune cells circulating in your blood, the B cell and the T cell. The T cell is basically for cell-mediated immunity. You hear some of this in the COVID literature. The B cell is the cell that makes antibody, the protein that is your primary defense against infectious disease. Your blood contains millions of B cells, and each one of those, when it becomes destined for a particular antigen, whether it's an infectious agent or anything else, will be permanently programmed to make only that one kind of antibody. So if you were to look at your entire blood, these millions of B cells are making millions of different kinds of antibody. What a monoclonal antibody does is to select one of those, to fuse it with another type of cell that will immortalize it and cause it to divide itself infinitely, and it will then produce what it's programmed to produce, which is this one super sensitive, super specific type of antibody. So if you think about having millions of different antibody molecules circulating in your blood versus a monoclonal antibody that is one only, if you get the specificity right, it is a truly potent thing. And that's what Synagis is, and that's what all these other monoclonal antibodies are that you hear about if you're watching TV at all. Synagis became an FDA licensed drug in 1999. As Tyler said, since then, about 250,000 high-risk infants, mostly very small premature infants, 32 weeks gestation and under, are the recipients of it. Others that have congenital heart disease or bronchopulmonary dysplasia also qualify for it. It's been highly effective. It reduces hospitalization by over 80%. And as I alluded to earlier, more than 2 decades have elapsed. The only other monoclonal antibody that is being used against any infectious agent right now is against COVID-19. And there's an asterisk by that, because that is not full FDA approval. That's only emergency use authorization during the pandemic. Now, lessons learned, at least that I've learned. I hope you'll learn some of them as well. Try to find the intersection between your passion and your toolkit. If you can even do that much, you're ahead of the game. You're ahead of what most people will do in their careers. And you will have a career that is both interesting to you and satisfying. If possible, work on something that's important. Whether it's your vocation or your avocation, choose something where you're likely to make a difference. There are plenty of important things around that need to be done. There's no shortage of them. Find them and do them. Number 2, follow the data. Make sure that you have trustworthy data and then go for it. And don't worry about the fact that you might be swimming against the current. Number 3, in an era where if it's not Googled, if it's not in Wikipedia, some people think it doesn't exist. Don't be afraid of diving deeply, of going beyond the computer databases, because sometimes you'll find some real nuggets that can make a difference. If we had relied on just the surface search that normally had been done, we would have missed that Russian paper, would never have gone down this road. And just as a footnote, to this day, that Russian paper is not accessible online. Never has been. The only way we found it was the deep dive manually into the literature. Number 4, be patient. Even though, as these things tend to go, the timeline between discovery and licensure of that first drug was 22 years, that's not a long time for something like this to happen. So particularly if you're in the sciences, be patient. The developmental pathway is longer. If you're in other areas of technology, That can be a fairly short pathway. You don't need as much patience. Number 5, at an appropriate time, if you really are trying to commercialize what you're doing, regardless of what field it is, bring on strategic partners. You can't do it all by yourself in today's world. And when you get to that point, look for the best. Don't try to do it on the cheap. There's nothing more expensive than an inexpensive attorney or consultant. Get it right the first time. Number 6, don't assume that everybody, and perhaps anybody, will love you if you are a pioneer. Because if you are overturning some of their treasured assumptions, they won't like you at all, and they may even try to block what you're doing. If you can, ignore it and plow through. Have faith in yourself, have faith in the data, and go for it. You'll see time and time again, if you look historically at the examples of successful companies, most of them have gone against conventional wisdom to get started in the first place. And number 7, if you do succeed, give something back. You've got a campus here that has names on most of the buildings. Those are people who did succeed and they gave back. Now, your turn. We've got, what, till 20 after? So we've got 15 minutes. Ask me what you want to ask. Yes, and speak loudly.
Okay, can you hear me?
Yes.
Okay. I mean, it looks like the process was very long to get that developed, and I'm sure you had lots of discouraging days. I don't know. What helped you get through those hard moments of discouragement, or did you even have those?
Yeah, I had those a lot, yes. Just trying to get that surgical procedure down took me over a year. Trial and error, because what worked very easily in mice and lab rats didn't work with cotton rats. Now, cotton rat is not like any lab rat you've ever seen. It's a different rodent family. I talked to one veterinarian, and she said her synonym for cotton rats was popcorn. So just that one procedure was driving me nuts until I finally figured out how to do the surgery and get the animals through the post-operative period so that we could do the whole experiment on it. Yeah, you just have to sometimes take stock of it and say, am I on the right track? And if I am, then okay, we're going to get this thing out. And everybody goes through that if they wind up doing anything of significance. Yes?
Has your dental degree helped you in any way with this research?
Yeah.
How so?
Most important thing was how to do microsurgery. Yeah, it was, in a way, it was a difficult procedure to tame, but doing the procedure itself, it was no big deal. There were physicians in our lab at NIH who didn't have a clue. I could have written the whole thing out, put all of the materials and equipment out. They couldn't have done it. Yeah, it was really, really helpful because dentistry, it's basically microsurgery, a lot of it. Yes, in the back.
How have you followed the data in other areas of your life as well?
The books I've written, some of which made some people rather agitated. Probably the most important book that I wrote was the biography of David Mckay. That's a name some of you probably are familiar with. And there was a good news and a bad news reception to that book. What I did was to follow the data all the way through and let the data tell the story. The founder of the LDS Church once said that truth is knowledge of things as they were, as they are, and as they will be. And I've got an addendum to that, and not as we wish they would have been. But a lot of history is written that way, and I refuse to do that. I took the data, went with the data. Former church historian has told me several times, he says, any new general officers that came in to the hierarchy, I would sit down and say, this is the book you need to read because this explains how this church functions. One of the other church officers said in a private setting, I like that book, but I wish Prince hadn't written it because it explains how the sausage is made. Now, following the data has never taken me astray. May not make you popular, but it'll get you to the right answer, and I think that's important. And you see in today's political world, the disregard for truth is one of the great existential crises facing this country. To try to manufacture something as your own brand of alternative reality is a dangerous road to go down, but we're seeing it wholesale, and it really scares me. So it makes me cling even more to the notion, follow the data, figure out what's true and cling to it. Yes?
You get a lot of pushback from other scientists?
Yeah. Yeah, even my own lab chief, Dr. Chanik. Now, this is the guy who discovered the virus. He's the guy who designed the vaccine that blew up. So here I am working in his lab. Not once did he ever walk into that lab. He just stayed in his office all the time. So he didn't even know what I was doing on this stuff. But I thought, okay, I could go in early and have him probably tell me, don't do this. And when I finally went in and talked to him, I had a stack of data about like this. And he looked through it, and he calmly said, we may have to rethink our biases. But others outside of that laboratory never took kindly to it. I think there may have been a little bit of jealousy there. I think there was an aversion to having a dentist come in and do this. But yeah, if you want to be popular, don't do cutting-edge research in anything. Yes, Tyler.
So as far as maybe your three careers as an author, a dentist, and an immunologist, how have you managed your time across those different things? Were you practicing dentistry while writing books?
All of the above. I practiced dentistry part-time for 21 years, but I put food on the table. When I started at NIH, because I didn't have an MD, I had 2 doctorates. I had a DDS and a PhD. My starting salary was $20,000 a year. So having the dental degree made up the difference. I never practiced dentistry full-time. That's not where my passion turned out to be. I didn't know when I went into things that's where I would wind up. Doing the books, I fantasized early on, gee, I'll carve out 3 months sometime and just go to some location uninterrupted and write. I never had that chance. I learned to write in 5 and 10 minute increments, mostly between dental patients for that first book. Never had an extended period. For about a dozen years, I was doing consulting with SmithKline Beecham, which is now GlaxoSmithKline, in their vaccine division in Belgium. And I would write a chapter on the plane flight back from Brussels to DC, because I had everything in the computer at that point. I just had to learn how to manage these things, and by the way, have 3 kids along the way. Yeah, if it's something that you're passionate about, you're going to figure out how to do it. But you're probably not going to be able to multitask and do the same thing that other people are doing who are not multitasking. There are limits, and we all have to work within those. Does that make any sense?
Yes.
It's not formulaic. Nothing is. And anybody who tries to tell you it is hasn't figured it out themselves. Yes?
So I feel like nowadays there's a lot of disregard for sciences.
Say that again?
There's a lot of disregard for science. People don't believe in vaccines. And why do you think that is, and how can we overcome it?
It's one of the questions that drives me nuts, and I don't have a good answer. His answer, his question, in case you didn't hear it, what about basically the anti-vaccine movement and just a general disregard of science? I don't know how to fix it. I see it as one of the largest dangers facing humanity. Disregard of reality is going to take you to a very dark place. Now, if you want to have a discussion about vaccines, then let's have that discussion and have it openly and honestly and talk about biology and how vaccines aren't 100% foolproof, but when you compare them against the alternatives, vaccination has been and continues to be the most cost-effective means of improving human health ever, period. And yet now we're seeing that a third of the country doesn't want to take this vaccine. It drives me crazy. I worked in the same institute with Tony Fauci. I know Barney Graham, if you've done any reading about the mRNA vaccine, Barney and his group at NIH developed the technology for the envelope around the mRNA that made the whole thing feasible. I've worked with Barney for 40 years. I know these people. And I know the quality of the work that they've done, and it just drives me insane. But my oldest son refuses to get the vaccine. What do you do? And I said, Chad, I've worked with all of these folks for decades. He says, well, if you'd made the vaccine, then probably I would take it. Your question is right on, and I wish I had a good answer, but I don't. And some of it may just have to do with the way some people are wired, that there is an inherent distrust of progress. It's basically, on steroids, the divide between conservatism and progressivism.
Do you think media plays a big role?
Yes, absolutely it does. I think the biggest role in the anti-vax movement as it relates to COVID goes to one man. And that one man, because of his own decisions about this, resulted in the deaths of hundreds of thousands of people. To me, it's unconscionable, but there it is. And it has a big following, and I don't know how to turn that around. Yeah. Yes?
I was just thinking in the world of health and this business specifically, how you have essentially vaccines to help people. How do you balance a good business plan and being able to maintain profits and things while also caring about making sure that this really inelastic item is able to get to people for good cost?
This is a recurring, again, the question here really has to do with the ethical basis of the pharmaceutical industry. I'm rephrasing that. Is that basically what you're saying? To try to find that balance between serving the public good and having a profitable company. There's plenty of room within fairly wide bumper guards to do that, but we've seen time and again companies that have gone past the bumper guards and done things that to me are just totally unconscionable. Something as simple as the EpiPen, you look at the price history of that and you say, come on guys, what's going on here? A drug that I used to prescribe, it comes in a little tube, it's called Kenalog, which is a topical corticosteroid in a petroleum base. You use it on canker sores, very effective. Little 5-gram tube used to be about $7 when it was not generic. Years later, I got a tube of it for myself, and it was a generic version, same size. It was $50, off patent. So yeah, something's out of whack with some of these, but I don't know what the answer is. It's not as simple as saying, all right, we'll give Congress the authority to regulate prices on the pharmaceutical industry. That gets into a big can of worms as well. But it's a subject that needs to be in the public square, it needs to be debated, and it needs to be continually brought out into the open. And perhaps shaming these companies will get us into a better place.
We have time for one more question.
Okay.
Based on that, so the ethics of these pharmaceutical companies, jacking up the prices and that sort of thing, that can lead to a lot of people mistrusting them.
Yes.
And getting these vaccinations. So like, you can kind of see where that crosses over, and just the mistrust in pharmaceutical companies in general, and not wanting to be told you have to take a certain vaccine.
Yep.
Like, as Americans, we want to push back against being told we have to do a certain thing. And so
It's complicated,
it is complicated.
but with the COVID vaccine, really the economics have been taken out of it. This vaccine is available to all for free. The price gouging there is not going on. And we've still got the people who refuse to take it. We also have a company in Baltimore whose former CEO is my next door neighbor in Maryland that finally got cut out of the contract by the federal government because they kept screwing it up. They made several million doses of the J&J vaccine that were contaminated. Fortunately, those never got into patients. Some of the stuff hits really close to home. Thank you for your attention. I'm around for a couple hours if you want to call me and talk about things. I'd be happy to do so.